So we take the last one before the coffee break, and then going back to the joint session. And this one is on stability, and the session lead is Matt. So please, thank you.
Yeah, thank you. So for saving time here, we go directly. Andrew Lennard will do the first presentation on application of predictive stability models to extrapolate shelf life, and then we will have a short discussion afterwards. Please, Lennard.
Thank you, Matt. So this presentation was done in collaboration with Biogen, UCB, and Roche. Next slide, please. Okay, so even for accelerated development programs, obtaining sufficient stability data from drug products is typically on the critical path. Furthermore, the lead time from data manufacture to batches being on the market restricts the remaining shelf life, since to avoid scrap and delay, PPQ batches are often planned for product launch. As a result, a comfortable shelf life for supply chain would typically be 24 months, with 18 months as perhaps the minimum feasible time. So here I attempt to provide some thoughts on how to reduce the likelihood of stability being on the critical path of biologics, with the common aim of faster patient access. So it's proposed that stability data for a biologic breakthrough prime product be extrapolated using mathematical models that are supported by predictive stability models that are generated from prior knowledge stability from structurally similar molecules. Such extrapolations take elements from the small molecule ICH-Q1E and reinterprets really the biologic stability of ICH-Q5C. And success of this type of approach requires that acceptance of risks in extrapolation of biologic stability data are appropriately mitigated by the prior knowledge stability. In addition, key milestones in development need to be locked down earlier than conventionally done. However, as we know, things happen, such as, for example, unexpected late reformulation of a product, and such unplanned events can result in significantly less stability data available for the breakthrough prime product. Next slide, please. Okay, so how does the proposed use of predictive stability model fit with the current ICH-Q5C guidance? The Q5C asks for primary data that are based on real-time data, and it could be argued that the model data from your prior knowledge is based on real-time data. Furthermore, you would have your stability batches for your breakthrough prime product as well. So data are expected from 3 representative batches. And again, you could see that your model will be composed of multiple batches, and indeed you would still have your 3 representative batches put onto stability, though at time of submission it may well be less than 3 batches. Containers are expected to be of representative material and type. And the minimum amount of stability data submissions is typically 6 months. Though I would say that when it comes to looking at stability and stability modeling, perhaps more important is just having a justified quantity of data so that you can have some confidence in the mathematical model that's being applied. So, and again, Q5C will discusses about setting clinical specifications, which, as we know, are generally expected to be clinically qualified, but we've heard about that in previous sessions. So, next slide, please. So, how might a predictive stability model be generated from the prior knowledge data? Clearly, criteria are required to justify that prior knowledge as being transferable to the breakthrough prime product, and considerations would include the structural modality of those molecules, the qualitative formulation that they have, the storage conditions, and indeed the container, as well as the analytical methods that are being used. And all these criteria should be allowed to have some flexibility in them as justified by the sponsor. So the prior knowledge should be analysed for similarity of trends per attribute. And since we are looking at the predictability of a trend, the exclusion of outlier products and normalization of the data should be considered. It is the trends that are particularly relevant for stability rather than the actual starting point. The sponsor should provide a rationale for any of these statistical analyses that are used, and the parameters are identifying goodness of fit. So those analyses will be both internal to the model, so you'll be looking at the similarity in your stability data across the different products that you're including in your model, and then also externally to the breakthrough prime product. But because predictions may not always be 100%, we should be able to consider some regulatory mechanism that might allow switching of the model that's being applied. And important to the generation of the predictive stability model and also its application in breakthrough prime would be the enhanced interactions with the agency, so we can get advanced agreement on both the forming of the model and how it's applied upfront by discussions with the agency. Next slide. So similarly, application of the model would also require predetermined criteria, because what you'll be doing is you're mapping the model onto the mathematical extrapolation of the real-time, real-condition breakthrough prime stability data. And a goodness of fit analysis should provide support that the extrapolated stability is appropriately supported by the prior knowledge data. Okay. So the conclusion of the analysis should also be put into the context of your product knowledge and also in the whole risk-benefit of the product for the unmet medical need. Furthermore, the proposed modelling would, of course, be verified on a continuous basis as product stability data is obtained and backed up by commitments similar to those we have at the moment for clinical studies in reporting new trends, out of specification results, etc. Next slide. So some example data are provided to illustrate what typical results may look like. These data are from the IgG modality that are all in a highly representative container, formulation and analytical methods used. And selected here some attributes that are known to be stability indicating. Only one attribute is illustrated here for frozen drug substance, since we do find that no attributes actually change for drug substance that's frozen. The product data are reflecting the assay variability, and the breakthrough prime product here in this example has got 12 months data. It's illustrated with a dash line. Overall, we do find that the risk for extrapolation of a drug substance could be considered as very low. For many drug products, formation of high molecular weight species that are predominantly dimer is often the primary degradation pathway, and typically follows exponential kinetics that plateau to a maximum level of dimer. The breakthrough prime product data are lying amongst the prior knowledge IgG data that are extending out to 36 months. The data support the extrapolation of the breakthrough prime stability data to at least 24 months, which in this case would be a twofold extrapolation. Though I would point out that if the breakthrough prime product had 6 months data available, then we would say that the model would still remain supportive of a 24-month shelf life. Next slide. So the similar data here are shown for cation exchange, acidic and basic peaks, and also for potency on the next slide. Okay, and on all the data in the selected model support at least a 24-month shelf life claim. Next slide. Okay. So, but what could be done to remove stability from the development program critical path when there is insufficient prior knowledge available? This is especially relevant for small and medium enterprises or when a new modality is being developed. So proposals to support extrapolated breakthrough prime stability data could include any combination of the following, such as extrapolating accelerated stability back to the recommended storage conditions, as might often be done perhaps with a small molecule, providing additional stability time point data, which would improve the confidence that the attribute degradation follows the predicted kinetics and provides further assurance that any changes in kinetics are detected. What we would call enhanced regulatory commitments to provide stability data at agreed time points. This could possibly be through a risk management plan, perhaps safety updates, or could be even through a legally binding commitment such as a leg. That's perhaps something that could be discussed as to what mechanisms are in place for these types of commitment. Furthermore, extrapolation could be perhaps more proportionate to the total amount of supporting information. So, for example, if you have little prior knowledge to support your extrapolation, then maybe it could be allowed to have a more limited extrapolation to say twofold when combined with the regulatory commitments. So this brings us then just down to the next slide with questions that we could discuss. It really is asking whether agencies could allow the use of predictive stability models for biologics to extrapolate the stability. When you have less data, perhaps more likely from less batches than normal, and could the absence of prior knowledge be supplemented by concepts such as accelerated stability modeling, additional time points and commitments, all in consideration of the risk benefit for the breakthrough prime product. So yeah, next slide, please. So yeah, it's just their final acknowledgements, really, from members of Amgen, Biogen, UCB, and Roche. So, thank you.
Thank you, Andrew. And we will now go into a short discussion, knowing that we are 2 minutes over time already when starting. So, unfortunately, but for the panel, we have Emanuela and Laurie from CEDAR and Andrew from CBER. In addition to Andrew, we also have Markus Goese, who is the lead EU CMC regulatory policy for Roche Basel and also chair of the EB Biomanufacturing Working Group. So I think if I just start, I think you have 2 really, really good questions, and which would deserve a meeting of this case on its own. But I just I think you point out to some interesting stuff here, that what matters is the slope, it's the trend, not really the results of the different batches, but really what do they look like, and then you apply that on whatever would be the clinically qualified specification or acceptance criteria. I think another issue, as you raised, is you have cases where the model doesn't fit. I think it would be very interesting or very useful to realize and to see why is that.
Yeah.
Because then that could justify your justification of saying this will fit the model. Because it doesn't have any of these aspects as the non-fitters have. So maybe some just...
Yeah, so I can say certainly for one of those examples, I did look back and talk to some people in that product team, and there was an unusual extra additional event that occurs with that product. In fact, talking to some of our people, it's the only time they've ever seen high molecular weight species increase linearly over a long period of time. And that was related to a clipping event which we've only ever seen with that one product. So it was an interesting sort of example to put in there to show how your product knowledge, and indeed I should say your accelerated data, can actually pick out atypical data that atypical products that you may have in your lake of prior knowledge.
Yeah, that's good. Sean, do you have
No, for me, I mean, the principle I think is acceptable in although this is, amongst ourselves, it's like a rubber stamp that this is not the way to go. But for me, the key would be, as you said, how does it fit into your model? And one possible way of that is the stress data, where you show that when we degrade it forcefully, it does behave in the same way as these other ones. So that gives credence to that. If the degradation behaves in the same way, then it should probably fit in the model. So, and that is one useful way. Because if you just have maybe 3 months stability data at the recommended storage, how do you know definitely that it's going to fit within the model?
Yeah. So I did try to make a point of the important thing isn't so much of having a fixed amount of data in terms of time, it's the amount of data that you have that gives you confidence in the model, the mathematical model that's being applied.
So can I just ask a clarifying question? What are the criteria of what sort of fits into the modality sort of bucket? Is it you have the same cell line, the same manufacturing process, it's a platform process? I mean, what sort of criteria do you have that says this product can be used in this model?
Yeah. I mean, so at the moment, it's being looked at quite a sort of high level, and it really is the structure of the molecule, the sort of modality and the structures that are present. A very simple one in monoclonal, you've got your FC domains and your Fab fragments as examples. But then we've got other modalities that are more elaborately constructed that are still in that bucket of monoclonal antibodies. But those would typically be considered as a separate modality because they are structurally different, even though they are classified as monoclonals. But yeah, I mean, it's like everything else with prior knowledge. You can bury yourself in criteria, and the more you do that, the more you lose your prior knowledge. And there's more criteria, and the more strictly you apply criteria, you have this horrific attrition rate in your prior knowledge.
Yes, the other side of that is, though, that sort of impurities can sort of alter degradation and things like that. So there's maybe some criteria around the manufacturing process and understanding what impurities could be in the final product.
Yeah. So you could have those types of criteria, but ultimately, perhaps we might say it was the molecule and the properties of the molecule that is perhaps more than the process. But that's just an opinion. And I'm not saying I've got all the answers here. Clearly, there are hurdles that would need to be overcome. But these are suggestions for further development and discussion.
I think the other issue you'd brought up, where you don't have the platform, you don't have the model, it makes it even much more difficult, because there, how to do it, I guess, again, coming back to some accelerated data, which could, but how much you can, much you can do there, because we have for clinical trials materials, yes, we could allow prolongation without having seen the data, but then all these batches are in a controlled scenario. So if something happens, it's easy to withdraw them. This will not be the case for these guys. So I don't know if anyone have any experience on how this can be done, because it clearly has to be done, but it's more difficult than this one. Marcus?
Well, maybe just a brief comment. I think you mentioned it already. I think there is tremendous value in these stress data, and we have also experience with this, looking at different monoclonals, etc. And I feel it's a little bit underestimated how much value is in these data still nowadays, and it can also be obviously helpful in cases where you are lacking a bit of this kind of experience, and then you just you go for it and look at 40-degree data, for example, 2 weeks, and it will be very interesting to look at these results and helpful.
I think it's fair to say we're a little bit hampered by Q5C being written in 1995. Things has happened, so the standard may also be different, and then if we're going a little bit further than that, we would be even further. But I think this is really issues that, and I think if we should have the time to have any coffee, I think we need to break here. But this is clearly an interesting topic, and if we would ask for full-time data, we will really sort of wait 2 years before you come on the market, and that is not acceptable to anyone, I think. So thank you very much for a good discussion.